Now let us know about the mechanism of vision. How actually we are able to see the things. Now the types of vision, if you take up, there are two types of vision we are making. One is phototropic vision and another is scotopic vision. What is the difference? Phototropic vision is the vision in daylight which we see in bright light. We are able to see everything in daylight or in bright light, and that is called as phototropic vision. And night vision, or in the dim light, if you are seeing something, or during the twilight hours. Twilight hours is the time after the sunset immediately and before nightfall. So there is a very little light around. So that kind of surroundings, we call them as we call such surrounding as twilight. So dim light is there. In that low intensity of light, the vision we are making, the thing we are able to see, that type of vision is called as scotopic vision. Okay. Now for these we have receptors, which are there in the retina, as we have studied. Rods are the cells which are important for the vision in the dim light or in the twilight. When there is a less light, we use our rods. Rods help us in seeing the things, but they cannot help us in detecting the color. So no color vision here. Only we can see the shades of black and white, black, white and grey. So that's why you might have experienced, if you are walking on a road in twilight hours and there is no street light also, it is the almost darkness is appearing. So you see that the things around us will be appearing either grey, white or black in color, because in dim light our cones, which are important for color vision, cannot function. In dim light only rods can help us to see the things. So therefore we can't see the colors, but we can see the things around. So that is for the night vision. Rods we have, and then the cones, which bring about the color vision. And the vision, color vision, is possible only in the bright light, either sunlight or any other artificial light. In bright light we can see the colors with the help of cones. Actually in our retina, in each eye, there are about 125 million rods and 75 million cones are there. And what about the distribution of these two? Distribution of the rods and cones, if you take up, distribution, rods uniformly distributed all over the retina, whereas the cones are restricted to the fovea and macula lutea. So that is how the two types of photoreceptors are distributed in our retina. And the shape of the cells we see here, rod has got a rod-like structure, therefore we call it as the rod cell. And inside the cell there are lamellae, parallelly arranged structures, and in these lamellae there are the photoreceptive pigments. So photosensitive pigments also we can call. So those photosensitive pigments found in the rods, they are called as rhodopsins. Rhodopsin is a pigment found in the rods. And in case of the cones we have iodoxin. Cones, they are called so, or cone cells, because their structure is cone shaped. Okay, and both the cells are connected to nerve ending, connected to neuron. The neurons have their nerve endings on the foot region of these two cells. Okay, so in both there are lamellae and they contain different pigments. Rhodopsin in rods and adopsin in the cones. And cones are of different types based on which color they are detecting. So cones which are detecting different colors, they are having different pigments, so their pigments are differently labeled again. What we see that there are totally like red, blue and green. These three different colors can be detected by three different types of the cones. Three different types of cones. One for collecting or the detection of the red color, one for blue and another for green. So they are differently labeled also, like erythrolate for the red color, about rods we have already studied. Let me rub it. Erythrolate, then blue, cyanolate, and green, chlorolate type of pigments we have. Right, three types of iodoxin. So these different colored pigments, they detect different colors. Okay, so therefore three types of cones are there. By combination of these three colors in different proportion, or in other sense, the number of different types of cones excited and their combined effect will help us in detecting the different colors what we see in light. And it is really amazing to know that about 80 million different shades of colors we can detect with the help of our cones in the retina of our eye. So that's about the cones. And let us see how these are connected to the nerves further. Now we see that this is the retina. Retina has got a basal layer, an epithelium, which is made up of pigmented epithelium. Okay, and next layer is attached to these pigmented epithelium, which have got microvilli also. So fitted into that you have rods and cones. Both rods and cones are first connected to bipolar neuron, and then they are having the synaptic connections, okay, with the ganglion cells, and these ganglion cells are further connected to, or they have the synaptic junction with optic neuron. So all the optic neurons together, they will form the optic lobes, and in between the bipolar neurons there are some connecting neurons, transverse neurons, which are like amicron cells, right? They bring about the interconnection between the different bipolar neurons. In this way, the excited rods and cones, when they are stimulated by the light falling on them, right? So they will send the sensory impulses, visual impulses, through these neurons and ultimately reaching to optic nerve, and optic nerve carries it to the central nervous system for further analysis and processing of the vision. Now what changes actually take place in the rods and cones, let us say, when the vision is brought about, the mechanism of vision. So what happens is when the rhodopsin pigment is stimulated by the light, it's a photochemical reaction therefore. So what happens is when the light strikes onto the rods, the rhodopsin splits into retinal and opsin. Retinal and opsin. Opsin, when it gets separated from the retinal, some conformational changes take place, its shape changes, and that makes the changes in the cell membrane of the rods. So that changes the permeability of the membrane, and this change in the membrane permeability will trigger or will generate a kind of impulse or a signal, and that is carried as electric signal by the neurons, bipolar, and from the bipolar transmitted to the ganglion cell, and from there it is transmitted to the optic neurons, and that's why the visual impulse ultimately through the optic nerves reaches the central nervous system. In the central nervous system, it is first received by the optic lobes, or we have as corpora quadrigemina we call in humans, and where the visual impulses are further processed and then sent to the vision center, which is located in the occipital lobe of our cerebellum. Occipital lobe of our cerebellum, that is the anterior part of the brain, cerebral hemisphere is what we have. So this vision center is located in this region. So actually in the vision center, which is at the back, the actual image is recognized. We see actually through the vision center in the occipital lobe of our cerebrum. So that is how the mechanism of the vision is brought about. This is about the changes what we see in the rods. Similarly in the cones also, as the cones are excited, different types of cones are excited, and the different types of the colors are being detected, and those signals arising from the cones will be sent through the optic nerve, and again the same way it is processed and analyzed by the CNS, so that we are able to see the or have the color vision also, right? So this is how the eyes are helping us to have the perfect vision. So with this we conclude this topic. We will take up some other topic.